Hi, my name is Brooke Brothers. I'm a senior in the Immunology and Medical Microbiology program here at WVU, currently working under Dr. Yvonne Martinez at the WVU Cancer Institute. And my research project investigates a long non-coding RNA, or lincRNA, called linc-SPRY3 and its role in cancer cell radiosensitivity. So non-coding RNAs are a type of RNA that, unlike mRNA, don't get translated into protein, but rather they remain as RNAs that have been found to play many regulatory roles in biological pathways. One such role for the linc-SPRY3 family is the competitive binding of a protein named IGF-2BP3, which is known to normally bind mRNA for the oncogenic proteins c-Myc and HMGA2. This competitive inhibition reduces the synthesis of c-Myc and HMGA2 in the cells, shown in preliminary figure 1, and this, as a result, increases the cell's apoptotic activity. And in cancer cells especially, apoptosis is an important mechanism, sometimes referred to as programmed cell death, that frequently gets dysregulated so that when they undergo treatment like radiotherapy, the cells will continue to proliferate rather than be able to recognize the radiation damage and die. One major aspect about cancer cells is that by the process of mutation, it's very common for their chromosomes to get spliced, duplicated, or even deleted. The linc-SPRY3 family I'm looking at is encoded on the Y chromosome, so only people who have a Y chromosome can express this lincRNA. We've previously found that in male lung cancer cell lines that express the Y chromosome, the expression of linc-SPRY3 increases after treatment with radiotherapy. And in male cells that have lost their Y chromosome, as expected, there's no expression of the lincRNA before or after radiation, seen in preliminary figure 2. Furthermore, cells with the Y chromosome and expression of linc-SPRY3 had higher apoptotic activity and sensitivity to radiation, while cells with loss of the Y chromosome were determined to be radioresistant with low apoptotic activity. The project I'm working on hopes to confirm similar findings in other male cancer types, currently including HPV positive and negative head and neck cancer, as well as glioblastoma, a type of brain cancer. 2 cell lines per cancer type, with or without the Y chromosome, were cultured and treated with an 8-gray dose of radiation. And 72 hours later, PCR amplification was performed to check for expression of linc-SPRY3 in the cells. In the HPV positive head and neck lines, linc-SPRY3 had an average 6.85-fold increase in the irradiated Y chromosome expressing UMSCC104 line compared to the non-irradiated control. And no expression was found in the Y chromosome lacking UMSCC47 line with or without radiation. In the HPV negative head and neck lines, the Y chromosome expressing Cal27 line had an average 53.41-fold increase with radiation. And the HN31 line that we thought didn't have the Y chromosome actually had a 3.0-fold increase with radiation as well, suggesting there might be low but mixed expression of the Y chromosome in this cell line. Lastly, for the glioblastoma lines, the Y chromosome expressing T98G line had an average 11.96-fold increase compared to the non-irradiated, while U3020s, thought to be missing the Y chromosome, did have an average 1.77-fold increase, which isn't high enough to be considered significant, but it also doesn't conclude absence of the Y. As it's not a very common thing to isolate and characterize a male cell line based solely off of the presence or absence of their Y chromosome, it's not very likely that we'll be able to obtain perfect controls for our experiment groups where 100% of the population in a cell has lost their Y chromosome. We plan to continue searching for better control lines and expanding into different cancer types, as well as perform duplicates for the data gathered so that we've established a strong foundation of preliminary data. After that, we plan to conduct assays to determine the cell line's apoptotic activity to further confirm the correlation between expression of linc-SPRY3 and cancer cell radiosensitivity. These findings could ultimately aid in the testing of male cancer patient samples to determine on a case-by-case basis what types of treatments might be the most beneficial and effective. Thank you.